Okay, so the real question now is, if we want to do something about Parkinson's disease, I think we have to figure out what synuclein is doing. What is it actually doing to our brains? It is a normal protein. But what's thought is that in Parkinson's disease, it may increase in some abnormal way and do something that's not normal at all, such as aggregate. And these Lewy bodies are aggregates of synuclein. You get other types of aggregates of synuclein. And somehow it's been thought that the aggregation of this protein, it's like clogging up things inside the nerve cell, actually produces the disease. But it's also possible that simply increasing synuclein by itself without actually getting these aggregates actually is responsible for the disease, and we really don't know. It's even possible that these aggregates, the Lewy bodies that people have looked at for many years, are a way for our brains to deal with having too much synuclein, and they're a way to kind of protect against it. If we didn't have them, things might actually be worse, because you'd have even more synuclein floating around. So one of the things we've been interested in, and one really basic question, is what does this protein normally do? It's a protein in all of our brains, and what does it normally do? What happens in Parkinson's disease? These are central questions. So now I'm going to get a little bit more into the scientific side, but we will come back to hopefully some of these bigger questions. What does synuclein do? Well, synuclein normally in our brains, in our nerve cells, it resides at the synapse, which is the site of communication between cells in the brain. In particular, there's a presynaptic element and a postsynaptic element. The presynaptic element releases transmitter. The postsynaptic neuron responds to the neurotransmitter. And synuclein is specifically expressed on the presynaptic side, where the transmitter is released. So we have a lot of clues as to what this protein is doing because it's in a place which is specialized for the release of neurotransmitter. And those transmitters include not just dopamine, but basically all neurotransmitters. Synuclein is not just present in dopamine neurons normally, it's present in all our neurons. So that's probably why it can accumulate in so many different types of cells. So it's in the site of release. And so it suggests, well, maybe it has something to do with the release of neurotransmitter. And that's one thing I'm going to get to. Another thing is, what happens to synuclein? How does it behave in our nerve cells? It's actually a little protein that floats around. If you just make the protein, it'll float around in a test tube. It'll float around inside cells, we think. But it's exquisitely located at synapses. So one of the questions we wanted to know is, since this protein is so important for Parkinson's disease, how does it behave? Let's just look at what the protein does. Does it sit in one place at the nerve terminal? Does it move around? Does it aggregate? What does it do? So these are the kind of experiments that my lab does, is we actually look at live nerve cells, dopamine neurons, glutamate neurons, all kinds of neurons in culture. That is, we take them out of the rat or the mouse brain. We look at them in culture. We look at the behavior of living cells. So a lot of work that's been done on synuclein has been done in a test tube. A lot of work that's been done on synuclein has been done with sort of artificial systems. We want to go back and figure out what's really happening in live cells because, frankly, that's what matters. So what we've done in this experiment is we have actually put a little fluorescent probe so we can see the synuclein protein. Of course, it's invisible. It's a protein floating around in cells. How can we identify it? So what we did in this case is we took a little fluorescent tag and we fused it to synuclein. We expressed it in cells, and we looked to see, well, we can follow the fluorescence. We can watch the synuclein move around in cells. And what do nerve cells do? They fire. That is, they have electrical activity. And the question is, with electrical activity, what happens to synuclein? So this just shows some live cells that we imaged. And these are synapses here, these yellow spots. This is the fluorescent synuclein at rest, sitting in the cells. And what we do then is we stimulate them. We cause them to fire the way the nerve cells in our brain are doing all the time. And the question is, what happens to the synuclein? And what you see here is that the synuclein gradually disappears from these synapses. So what this says is that a lot of activity causes the synuclein to move away from its normal location, and very slowly it moves back. What does this mean? We don't really know exactly yet, but it says that activity influences the behavior of this protein which causes Parkinson's disease. So what we'd like to think, and we'll come back to this at the end, is that the way it's when people think about the environment as causing diseases, like exposure to toxins, other sorts of things, you know, it's been estimated, you know, we've all heard about some of the stories, some pesticides and others can predispose to Parkinson's disease and other things. But what I'd like to get you to think about is not just the external environment in the world, but the environment in our brain, the activity of the cells, the way the circuits behave. What they do is going to influence the behavior of this protein, which causes Parkinson's disease. So activity, we think, is going to have a really important role. Something about the behavior of the circuits, that environment, the environment in our brain for this protein, is going to be important for this disease. Exactly how, we do not know. And so what we have found by imaging in cells is that synuclein, so what these are here, these green things are synaptic vesicles filled with neurotransmitter. The way all synapses in our brain work is these little vesicles come up to the surface of the cell here, and then they fuse and release the green stuff, which is the neurotransmitter. Okay, that's how all our synapses are working. As you're listening to me now, this is happening, and the neurons, you know, hopefully are awakened, so more of them are working. So that was good. Basically what we found by doing these kinds of experiments is synuclein is a protein that loosely associates with these vesicles, which are called synaptic vesicles that release the neurotransmitter. It comes on and off. And basically what happens, I keep losing my arrow, is that when the vesicle fuses with the plasma membrane to release the neurotransmitter, the synuclein comes off at that step. So in other words, we can do these experiments, we can do them very precisely, and we can find out exactly when synuclein is coming off, and we can manipulate the system to figure this out. There are other proteins that behave, that also disperse from synapses with electrical stimulation. But synuclein is different in that when it comes off and disperses, it comes back unbelievably slowly. So, okay. So, okay, some more bigger ideas again. One of the big questions is whether the disease is caused by a gain of the normal function. We know that an increase in synuclein causes Parkinson's disease. The question is that because it's increased in its normal function, or it's got an abnormal function like aggregation. And I probably should not get into some of the technical details here, but there are various models of toxicity which have been shown to be changed by getting rid of synuclein. And what this says is that the normal function of synuclein is probably very important for certain forms of toxicity. So what I think is, and it's not completely proven, is that this disease, unlike many other neurodegenerative diseases like Alzheimer's disease, is probably due to a gain in the normal function of this protein. It's not completely proven yet, but so the question is, what is the normal function of this protein? Okay, so this is some other types of experiments we do in live neurons. It's very simple in this case. What we do is we take these nerve cells from rats or mice, and we image the actual fusion of synaptic vesicles. So this green thing here, this is the green thing. When it's inside the cell, it's not green, it's not fluorescent. With exocytosis and release of transmitter, it becomes more fluorescent, and then when the vesicles recycle, it becomes less fluorescent. The bottom line is we can monitor things that you can't even see them really with the microscope, but by using certain tricks, we can actually monitor very precisely the behavior of these synaptic vesicles in the nerve terminal, and then see what does synuclein do to them. What I showed you before is what activity does to synuclein. Now what we're asking is, what does synuclein do to the activity of our nerve cells? And basically what this shows is here in black is when we stimulate here, 10 hertz, that's stimulating 10 times per second for a minute. And what you see is the increase in fluorescence is the release of neurotransmitter. The decrease in fluorescence here is when those vesicles come back in. So we can monitor both of those. When we overexpress synuclein here in red, you get less transmitter release. That's the bottom line. And what we've done with lots of experiments is synuclein, and this is without aggregates, without obvious toxicity. We think this is the first thing that synuclein is going to do when it goes up. It's going to inhibit neurotransmitter release. And we think that's its normal function, but when you make more of it, it does more. If we make enough synuclein in cells, we can eliminate transmitter release. So the normal function doesn't sound so good. Of course, in our brains, we have things that make synapses work better and worse because you have to modulate them to get our circuits in our brain to work properly. This is one that's normally involved in that. But now if the levels go up too far, it can kill the activity. So this is more ways that we've shown it. There are a lot of molecular details about how this works, and we haven't worked all of these out, but I think there are lots of different ways of looking at this. I always wonder what I was thinking when I put all these slides in. But this is the bottom line. There are actually different pools of synaptic vesicles in the nerve terminal. Let's say at most of the at a given synapse, maybe you have 100 of them. Now, I should say that synapses are far away from the central part of the cell where the cell produces things. They're really far away. So they kind of have to operate autonomously for days to weeks to months. So these synaptic vesicles, you have only 100. You probably release all of them that can be released in a minute or two, but they're out there for weeks or months, so you have to recycle them. So that's the point. And actually, of all the synaptic vesicles, maybe you have 100 in a nerve terminal. The blue and green guys are the ones that do all the work, and they can be only 10 to 20% of all the vesicles there. Do all the work. The other 80%, we don't really know what they do, but they're sitting there. And that's the normal situation. And when you overexpress, so these blue and green are the ones that are doing all the work. When you overexpress synuclein, basically, they go down. The total number of vesicles doesn't change, but the ones that can release goes down, and the ones that are just sitting there increase. We think that it's because they're actually spread further away. We can actually take pictures in an electron microscope and show that the vesicles are further away from the site at which they release. So they're spread out here as opposed to being more clustered over here. That's what we found. So we have molecular clues as to what's going on, which we're interested in pursuing. And I think in the interest of time and avoiding some of those details, I want to focus on the big points here, which is that synuclein normally functions to inhibit the release of neurotransmitter. When it goes up in Parkinson's disease, it's going to inhibit it more. And we think this is the normal function of the protein, but there's just too much of it, and it's not good when it goes up that high. The key thing here is that what we're finding, these are the earliest changes in Parkinson's disease. What we think is Parkinson's disease is the levels so some people have an extra copy of the gene, some people have a mutation, but most people with Parkinson's disease don't have any major hereditary predisposition to it. The protein just goes up. We don't really know why it goes up. It could go up because it's not being disposed of. It could go up because the cell's making more for reasons we don't understand. But it goes up. And before you get aggregates, and before you lose cells, and all the things that we hear about, which are kind of scary because they're irreversible, before any of those things happen, synuclein is impairing the function of those synapses. And what that says is there's clearly physiological changes that occur before the loss of cells. And that's actually kind of hopeful, because what it says is we could intervene at that point and get those cells to work better before they're actually lost. So that's one of the hopeful things. It probably also helps to explain some of the prodromal symptoms in Parkinson's disease. Often patients have constipation, trouble with smell, sleep disorders that can precede the disease by 10 to 20 years. I would say that actually those disorders are probably not due to the loss of neurons in the brain, the way we think of Parkinson's disease usually. I would say that those are functional defects, which could be reversed if we could make the synuclein levels go down or stop what it's doing. And I would even go so far as to say, we all hear that to get the motor symptoms of Parkinson's disease, you have to lose 50 to 80% of the cells of the dopamine neurons. And that's true. To develop symptoms, you have to lose a lot of cells. But those 20% or 50% that are left, I would venture to say that they're not working right. And if we could even take the cells that are left and get them to work better by lowering the synuclein levels or, again, blocking what synuclein does, people might, you might have more dopamine neurons, functional dopamine neurons than we really appreciate. And if we could reduce, even for people that have had the disease for a while, it could have a huge effect. So,